Nervous System Structure and Function Lecture. The objectives for this lecture are to list the organs and the division of the nervous system and describe the generalized functions of the nervous system as a whole, identify the major types of cells in the nervous system and discuss the function of each, identify the structure of a nerve, identify the anatomical and functional components of the Three-Neuron Reflex Arc, compare and contrast the propagation of nerve impulses along the nerve fiber and across the synaptic cleft, and identify neurotransmitters related to disorders. Organs and division of the nervous system. The nervous system is divided into two major parts. There's the central nervous system, which consists of the brain and the spinal cord, and then there is the peripheral nervous system, which is all other nerves outside the brain and spinal cord. The peripheral nervous system is then divided into the autonomic nervous system, or involuntary nervous system as we call it. And this autonomic nervous system is also divided into sympathetic and parasympathetic divisions. Here is a chart on the divisions of the nervous system. So the central nervous system relays messages between just the brain and the spinal cord. The peripheral nervous system has spinal nerves and cranial nerves. As you see here, cranial nerves respond back to the brain directly, and spinal nerves respond back to the spinal cord, which then relays the message to the brain. The autonomic or involuntary motor nerves and the somatic voluntary motor nerves, which is mainly of skeletal muscle, both report back to spinal nerves and cranial nerves depending on where they are in the body. Sensory nerves also report back to or relay the message back to the cranial nerves and spinal nerves. Our sense of smell, taste, and eyesight often are within the cranial nerves, which then relay the message to the brain. However, our sense of touch, which can be anywhere in our body, is generally relayed to our spinal nerves, which is then relayed to our spinal cord and back to our brain to receive the message. Cells of the nervous system. There are two main types of cells found in the nervous system. There's the neuron or the nerve cell and the glia, which is a specialized connective tissue cell. Neurons consist of three parts. There's the cell body of the neuron, which is the main part, and then there's the dendrites, which are branching projections that conduct impulses to the cell body of a neuron. The axon is the elongated projection that conducts impulses away from the cell body of the neuron. For your test, you will want to know the difference between the dendrites and the axons, and how I remember is axon is away, they both start with A, and dendrites are to the cell body. Here's a picture of the structure of a neuron. So A is the diagram of a typical neuron, and as you can see, the axons here. And then you have the cell body up here, and the dendrites are branching off of that cell body. B is a segment of a myelinated axon. So as you can see, the axons, and then the myelin sheath surrounds it in several layers, which the myelin sheath consists of many layers of Schwann cells. And then C is a neuron under a microscope, and what that looks like. Neurons classified according to their function or direction of impulse. So a sensory neuron conducts impulses to the spinal cord and brain, also called afferent neurons. Motor neurons conduct impulses away from the brain and spinal cord to muscles and glands. They are called efferent neurons. These efferent neurons only conduct impulses to muscles and glandular epithelial tissues. Interneurons conduct impulses from sensory neurons to motor neurons and are called the central or connecting neurons. Glia or neuroglia is, in Greek, glia means glue. So the glia support cells bring the cells of the nervous tissue together structurally and functionally. The glia cells hold the functioning neuron together, they protect them, and regulate neuron function. There are three types of connective tissue cells of the central nervous system. There are astrocytes, which are star-shaped cells that anchor small blood vessels to neurons. There are microglia, which are small cells that move in the inflamed brain tissue carrying on phagocytosis. And then there's the oligodendrocytes, which form myelin sheaths on axons in the central nervous system. Schwann cells form myelin sheaths in the peripheral nervous system only. And myelin is usually a white fatty substance. So here's a picture of a glia of the central nervous system, and as you can see, astrocytes have extensions attached to the blood vessels. So you have the blood vessel here, and then they have little extensions. In B, the microglia are enlarged and perform phagocytosis in the central nervous system. Oligodendrocytes have extensions that form myelin sheaths around the axon in the central nervous system, as you can see here. Nerves. A nerve is a bundle of peripheral axons. A tract is a bundle of central axons. White matter consists of brain or core tissue composed primarily of myelinated axons or tracts. Gray matter consists of brain or core tissue composed primarily of cell bodies and unmyelinated fibers. So here's a picture of the nerve. As you can see, we have the fossicle, which is surrounded by the perineurium, and inside this fossicle you have axons with myelinated sheaths, which consists of Schwann cells. They are surrounded by endoneurium, and there are several of these fossicles that can be within one nerve, along with arteries and veins and fatty deposits, and then is covered by epineurium, which consists of one entire nerve. Nerve coverings. They are fibrous connective tissues, and an endoneurium surrounds individual fibers within a nerve. Axons are then bundled together and they are called fossicles. The perineurium surrounds a group of fossicles of nerve fibers, and the epineurium surrounds the entire nerve itself. Reflex arcs. Nervous impulses or action potentials are conducted from receptors to effectors over neuron pathways, or reflex arcs. Conduction by a reflex arc results in a reflex. A reflex arc is the simplest type of neuron pathway in the body. There are generally two types of reflex arcs. There's a two-neuron arc, which consists of a sensory neuron synapsing in the spinal cord with a motor neuron. And then there is also a three-neuron arc, which consists of sensory neurons synapsing in the spinal cord with interneurons that synapse with a motor neuron. So here you can see a two-neuron arc, I should say, in the patellar reflex. So what happens is we stimulate the patella, and it causes a response via the sensory nerve back to the spinal cord here, and then it relays through the synaptic cleft in the gray matter a message, and it goes via the motor neuron back to the muscle to cause the actual response. So a simple two-neuron reflex arc, in summary, a stimulus generates a nerve impulse. The impulse travels down the sensory neuron's dendrite to its cell body in the dorsal root of the ganglion into the axon. The impulse travels into a microscopic space called the synapse between the axon and the sensory neuron and into the dendrite of the neuron in the gray matter of the spinal cord. In the synapse, the chemical signals are sent across the gap. These are known as neurotransmitters, and the impulse continues along the motor neuron. The motor neuron then forms a synapse with an effector, an organ that puts the nerve signal into effect, which causes our leg to move. A three-neuron reflex arc is a little bit more complex in response and involves an interneuron in addition to the sensory and motor neurons. The end of the sensory neuron's axon synapses first with the interneuron before chemical signals are sent across a second synapse, finally conducting through a motor neuron. Interneurons are located entirely in the gray matter of the brain and the spinal cord. Nerve impulses. The definition of a nerve impulse is a self-propagating wave of electrical disturbance that travels along the surface of a neuron membrane and is often called an action potential. As you can see here in the picture, so up here on A is an unmyelinated fiber. A nerve impulse is self-propelling wave of electrical disturbance that goes where it needs to. B is a myelinated fiber that the action potential jumps around, so it jumps between one to another around the insulating myelin in a rapid type of conduction called saltatory conduction. Nerve impulses. At rest, the neuron's membrane is slightly positive on the outside and polarized from a slight excess of sodium. On the outside, a stimulus triggers the opening of the sodium channels in the plasma membrane of the neuron. Impulses are initiated by pressure, temperature, or chemical changes. Impulse mechanisms. Inward movement of sodium depolarizes the membrane by making the inside more positive than the outside at the stimulated point. This depolarization is a nerve impulse or an action potential. They call these action potentials because each impulse has a different charge. The stimulated section of the membrane immediately repolarizes, but by the time the depolarization has triggered the next section of the membrane to depolarize, thus propagating the wave of electrical disturbances. Depolarizations all the way down the membrane. Saltatory conduction. When traveling impulses encounter a section of a membrane covered by insulating myelin, they jump around the myelin from one gap to the next. A synapse. The definition of a synapse is the place where the impulses are transmitted from one neuron to another, the postsynaptic neuron. Synapses are made of three different structures: the synaptic knob, the synaptic cleft, and the plasma membrane. Neurotransmitters bind to specific receptor molecules in the membrane of the postsynaptic neuron, opening ion channels and thereby stimulating impulse conduction by the membrane. So here's a picture of a synapse, and as you can see, we have the synaptic knob, the synaptic cleft, and then the plasma membrane of the postsynaptic neuron. A nervous impulse travels from one neuron to another through the synapse via a neurotransmitter. On the arrival of the action potential at the synaptic knob, which is here, a neurotransmitter molecules are then released from the vesicles in the knob into the synaptic cleft. So these are little neurotransmitters being released into the synaptic cleft. The combining of neurotransmitters and receptor molecules, so then the neurotransmitters are recepted by these little receptors here in the plasma membrane of the postsynaptic neuron, opens ion channels and thereby initiates the impulse conduction in the postsynaptic neuron. So then an impulse is then created and moves on. The synapse. Neurotransmitters are chemicals in which neurons communicate. There are at least 30 different neurotransmitters within our body. Some examples of neurotransmitters are acetylcholine, catecholamines, endorphins, enkephalins, nitric oxide, and other compounds. Acetylcholine is released at synapses in the spinal cord and neuromuscular junctions. Other neurotransmitters, such as catecholamines, and examples of catecholamines are norepinephrine, dopamine, and serotonin, all play a vital role in other functions such as sleep, motor function, mood, and pleasure. Endorphins and enkephalins are natural painkillers and are considered neurotransmitters. Parkinson's disease is characterized by abnormal low levels of dopamine in motor control areas of the brain. Patients usually exhibit involuntary trembling and muscle rigidity.